So hi, my name is Remi Olsen. I'm a bioinformatician, and I work mainly with anything that is de novo sequencing. So I will give a short overview over all the multiple methods we provide here at NGI Stockholm, and also the de novo assembly service that we provide. So first, I will talk about using regular Illumina libraries. We are an Illumina facility. We have Illumina sequencers, and we prepare Illumina libraries. So this is kind of the main workhorse of our de novo sequencing. Of our de novo sequencing. So first, we have two types of libraries. We have the TruSeq PCR-free, which with both Pat and others have talked about, and we provide this in several insert sizes. And secondly, we have the Nextera mate pair libraries with longer insert sizes. One type at around 3000 kb, one at 5 to 8, and one at 20 kb. So the idea of using these libraries is to follow what we call the all paths recipe. And as you can see on this log plot on this slide, it gives you the idea of what it's about. So the two first paired end libraries are used for config assembly. That is a contiguous sequence without any gaps, and then using the mate pair libraries in succession to bridge gaps in the genomes. That is, for instance, repeats that cannot be spanned by these shorter libraries. I will now talk about our bioinformatics pipeline for doing this work. It's called NuGet. It's basically 3 sub-pipelines. The first one is quality control. This is run by our production bioinformaticians on every project, and it does read trimming to trim low quality reads and adaptive read through. It runs FastQC and also plots the K-mer frequencies. I wish I had more time to talk about that. It's an interesting topic in itself. Second part is the assembly, and it supports a wide range of assemblers. We usually run 2 or 3 at a time for each project. And lastly, there's an evaluation pipeline. I will talk more about this on a later slide. So these are some example projects from 2016 that I just want to show you. It's you can see what type of organism it is and which assemblers that were run and which libraries, and also the N50 numbers for these assemblies. Now N50 is a measure of how well the assembly is connected, and a higher number is better. So as you can see, there's some you can't always predict how well a project will work. For instance, we have 2 birds. We have bird number 2 and bird number 3. They were sequenced using the same types of libraries and at approximately the same coverage, but the results are very different. I suspect that this was because of a faulty mate pair library. And for assembly evaluation, we have the N50 value. It doesn't always tell the full story. So we have several other measures, mostly in the upper left-hand side, we have what we call feature response curves. It's a measure of areas of suspected misassembly when we map the reads back to the genome assembly. And these areas are called features. So the way to read this plot is, if you're familiar with a receiver operating characteristic, it should be read in the same way. And secondly, we also run Busco. Shown here is using a avian dataset consisting of around 5,000 genes. So it's a set of it's a set of genes that it's evaluated against. It's single copy orthologs. So it counts how many genes you find, how many duplicates, and so on. And we see that the all parts assembly is the best one. It finds the most genes. Also, we plot the coverage versus the contact lengths, and we see that this is a diploid genome. All in all, taken together, the all parts assembly was the best one. So over to the 10x Chromium. Shown here is the workflow. It's long pieces above 50 kb that are encapsulated in droplets with gel beads and with the barcodes and necessary reagents. Yeah, at the end you will get a regular Illumina library with about 300 base pair inserts that we run on typically the HiSeq X. So what you can use this for when it comes to de novo sequencing is first of all the assembler from 10x Genomics called Supernova. So it has a very specific set of input requirements. It works best if your genome is sequenced to around between 38 and 56x coverage, and with a minimum of 50 kb input molecules. And it is mainly made for human or mammalian genomes. But of course, we have tried other genomes, which are, yeah. And the other method is to use these reads for assembly scaffolding. Currently, there are two programs for this. There's ARCS and there's FragScaf. So I have tested FragScaf for a HapMap sample. It's a SOAP de novo assembly combined with a 10x run that we did for this same sample. Of course, the Supernova assembly was surprisingly good for this sample, but at least using this method for scaffolding shows promise. And also ARCS is a program that is specifically made for this type of data, but I have yet to try it. So I will show you this plots from the first 7 human samples that we sequenced. This is showing you the scaffold N50 of the assemblies as a response to the molecule length, input length. So there was a point about this in the supernova paper, which I got the numbers for the red, marked with the red points, compared to what we did. And if you kind of squint your eyes, you will see that there is a threshold in quality around 30 to 50 kb input molecules. This is for 4 bird genomes project, 4 bird genomes that we did, and here the story is not the same. First, the 2 first ones in green and yellow, you might see this response, but then we have 2 more projects, especially the red one, appropriately marked in red, where something might have gone wrong. We are investigating it currently. Some more early results from our sequencing. These are genomes that fall well without the outside the recommended inputs for supernova, but we have run it anyway. Insects, a few marine animals, and so on. I will not talk too much about this. So moving on. I will not talk about our RADseq protocol. So as the name implies, RADseq is restriction-associated DNA sequencing. It is a method for giving a reduced representation of a genome. So typically you will only sequence between maybe 1% and 5% of the whole genome. This just gives a quick overview of our kind of homebrewed protocol for doing this. So a genome is digested, in this case using EcoR1, and we have adapters that fit to the overhangs of these digestion sites, and then we do a site selection and then sequence these libraries. For analysis, I usually use a pipeline called Stacks, and this is for working with this type of data without a reference genome, which is often the case. It will assemble in a way. It's not really a genome assembly. This reads into what they call stacks, and it will build a catalog of genomic loci and call variants, and also a sort of a population catalog as well. You can this slide is taken from their homepage, where you can read more about this. An interesting project that we are currently collaborating with Göran Spång at Umeå is the sequencing of African lions and leopards, which we have well over 200 samples now. Two interesting, or at least the interesting plot here is the on the upper right-hand side. It shows the number. I took out 24 lion samples and saw how many loci are shared between each sample. So it shows that this data set is quite connected and doesn't only consist of singleton loci, which would be really bad, and you couldn't really tell much from it. Next, I did a PCA plot from the variants that stacks outputs, and we see that on the first component, the two species are quite discriminated, and also the geographical population of lions, they also cluster kind of well with some outliers due to most likely due to very different coverage levels, but this might be some samples that could be taken out of the analysis.